A very warm welcome to all. So, my name is Charanjit Kaur and I am pursuing PhD in biochemistry from Punjab Agricultural University, India. And I have been working on sustainable agriculture and food processing from the last 3 years. And I'm quite interested to share some of the insights from my recent research work, which is valorization of carrot bio waste to bioproduct by ultrasound-alkali-mediated extraction. So we know as the world is grappling the challenges such as food security, the major issue which we are facing is the nutritional deficiencies. So among that, the protein energy malnutrition is one of the most alarming deficiencies which the people are facing. So kwashiorkor and marasmus, these are the common protein energy malnutrition diseases, and they can only be overcome if the recommended dietary allowance of protein is taken in their daily diet. So therefore, there is need to have various sources of proteins which are of good quality and they are also sustainable and accessible at the same time. So, according to a recent research, it has been seen that in 2022, the total plant-based protein market was around 12.1 US billion dollars, and with the increase of 7.3%, it has been estimated by around 2032 it will reach to around 24.1 billion US dollars, which is very huge. And it can be seen that the major burden is on the soybean, pea protein, and the wheat protein. And there is very less research on the other sources of proteins which could be easily utilized to increase this market potential. So these plant-based proteins, they are gaining huge popularity. So there are many reasons behind this. So first is because of the lifestyle choices such as being vegan and vegetarian, or increasing population, low economic and financial status of people, and high cost of animal based proteins, or the medical needs such as lactose intolerance or the presence of allergens in the animal based diet. Therefore, researchers and scientists, they are shifting their focus to increase the valorization potential of the underutilized crops so that the sustainable, accessible, and good quality proteins can be extracted from these methods. We know that carrot is one of the top 10 economically grown vegetable crop in the world. So, as I have worked in India, and according to the present data of India, the area under the cultivation of carrot is 1.4 million hectare with the annual production of 1.6 million tons. And we can see that the huge amount of leaves they are getting wasted, as around 0.48 million tons of the carrot leaves they are usually discarded or burned or underutilized. However, these carrot leaves they have huge potential. It has around 18 to 22 grams of protein per 100 grams of dry weight. It is very rich in carotenoids, polyphenols, and flavonoids. So, despite all of these nutritional benefits, there are some of the constraints due to which these carrot leaves they are not being directly consumed. It is because of the presence of high crude fiber and the anti-nutrients such as saponins, tannins, phytates, oxalate, and flavonoids, which are limiting their digestibility and thus the human utilization. Therefore, the objective of my study was to extract the leaf proteins using alkali and ultrasound alkali assisted methods with their process optimization, and then characterization of ALPCs, which are alkali assisted leaf protein concentrates, and UALPCs, which are ultrasound alkali assisted leaf protein concentrates for functional and biochemical and nutritional properties. So this was the methodology which we had followed. So the carrot leaves, they were procured from the fields of Punjab Agriculture Universities. They were sorted and washed and then dried at 55 degrees Celsius in a tray dryer, followed by grinding to fine powder and defatting the carrot leaf powder. This defatted carrot leaf powder was further used to carry out the optimization using two methods. So, one is the alkali assisted method and another is ultrasound alkali assisted leaf protein methods. So, in both the optimization methods, three independent parameters they were under consideration. For example, the pH, time and solvent is to solid ratio was taken for alkali assisted extraction, and power rate, time, and solvent is to solid ratio was under consideration for ultrasound alkali assisted extraction. After the ultrasound treatment of carrot leaves, second treatment was also given, which was the optimized condition of the alkali extraction, followed by homogenization, isoelectric precipitation, centrifugation, followed by the freeze drying of the leaf protein concentrates thus formed. These leaf protein concentrates, they were further studied for their biochemical, functional and nutritional properties, which are listed as follows. So this is the response surface methodology for the extraction of alkali assisted leaf protein concentrates formed by alkali extraction. So the box Benken method was used and 17 runs were made. So pH of 7 to 12 was taken and extraction time was from 30 to 60 minutes and solvent is to solid ratio was from 10 to 20. So two responses were determined, one is yield and another is the protein. So the major outcomes, they are highlighted in red. So we can see that the maximum yield which could be achieved was around 6.96% and the minimum yield which was determined was 3.14%. And the maximum protein content which could be extracted in the leaf protein concentrates was 44.18% and the minimum protein content achieved in the LPC was 12.78%. So we can easily see that the alkali assisted extraction is not a very good extraction as only 44.18% protein was extracted. This could be explained because plant cell contains only 20% of the soluble proteins which are present in the cytoplasm, and alkali extraction could only lead to extraction of these soluble proteins. Therefore, the protein content determined by this kind of extraction is quite low. Therefore, alternative and innovative approach has been used, which is ultrasound alkali assisted extraction. So this is the slide which is again showing the response surface methodology for the extraction of UALPCs, and the three independent factors were power rate, time, solvent to solid ratio, and the yield and protein content was the responses determined. So we can see that the maximum protein content which could be achieved after two subsequent treatments was around 68.55% and the minimum protein content achieved was 40.1%. There was also increase in the yield. So the maximum yield achieved was 8.66% and the minimum yield was 5.97%, which was at 30% power rate for 5 minutes and 15 is the solvent is to solid ratio. So the yield is minimum because the time of the extraction and the power rate is less, while the highest yield was at the 50% power rate, which could easily lead to the disruption of the cell wall membrane and even the chloroplast membrane, leading to high amount of extraction of the bound protein. So when the ultrasound treatment has been provided, so due to cavitation, so protein can be easily extracted out into the medium. So these are the 3D graphs for which are showing the interactive effects of two process parameters, taking the third parameter as constant. So from these graphs we can see that the quadratic model has been followed by the alkali assisted leaf protein extraction. So this is the optimized conditions which are listed for ALPCs and UALPCs. A very clear and significant difference can be seen between yield and protein content by both the methods. So after optimization, the functional properties of ALPCs and UALPC were determined. So we can see that the oil holding, water holding, foaming capacity and emulsion capacity had a significant difference, and UALPCs had high water holding, oil holding, foaming capacity and emulsion capacity. However, there was no significant difference between the forming stability and emulsion stability of ALPCs and UALPCs. So talking about the protein solubility, it was seen that both the ALPCs and UALPC showed increase in the protein solubility as the pH has been increased. The minimum solubility was at pH 2 while the maximum solubility was at pH 10. However, as the pH is increased to 12, the solubility of the protein has also decreased in case of UALPC. It could be because of the protein degradation at very high alkaline environment leading to low protein solubility. So coming to the biochemical properties of ALPCs and UALPCs. So after fractionation, we observed that glutalins, they were the major fraction which is present in the ALPCs and UALPCs, followed by the prolamins, albumins and globulins. There was a significant difference between the glutalins and globulins of UALPCs and ALPCs, while there was no significant difference among prolamins and albumins. After this, the antioxidant properties of these protein fractions was also determined. So we can see that in case of DPPH free radical scavenging activity, it was the glutalins which had maximum DPPH radical scavenging activity, followed by the globulins. In case of ABTS, again glutalins were responsible for the maximum ABTS activity. However, in case of FRAP, it were the prolamins which had the maximum FRAP activity. We also determined the anti-nutritional content present in ALPCs and UALPC, and the data was compared to that of the anti-nutrients present in the leaves. So it is very clear and evident that the significant differences there among saponins, terpenoids, tannins and alkaloids, and the UALPC had minimum amount of these anti-nutrients on comparison to ALPC and the leaf proteins. This table is again showing the percent decrease in saponin, terpenoids, tannins, and alkaloids. Clearly, UALPC had the maximum decrease in the anti-nutrient content in comparison to ALPC. So the nutritional profiles of ALPCs and ULPCs is also in front of you. So we can clearly see that all the 9 essential amino acid except for phenylalanine is present in the leaf protein concentrates thus prepared from the carrot, and the methionine, which is usually quite low in the leafy vegetable protein concentrates, it is present in case of carrot leaf proteins, so which is very different from the other studies. Moreover, the UALPC has high amount of amino acids on comparison to ALPCs, whether it could be aromatic, it could be sulfur containing essential amino acid, polar charged amino acids or non-polar amino acid. So we can say that it is nutritionally better than ALPCs. So on comparing ALPCs with UALPCs in terms of their in vitro digestibility, there was no significant difference seen. Talking about their secondary structure, the FTIR analysis of ALPCs and ULPC has been done. So amide 1 band could be clearly visible in both the graphs. The beta sheet content present in the ALPCs was 79.12%, while beta sheet content in UALPCs were around 74.44%. So in conclusion, we can say that UALPC production leads to increased yield, that is from 6.96% to 9.63%, and increased protein content, which is 44.18 to 66.72%. So it UALPC has been associated with better functional properties, antioxidant properties, and no compromise has been done in case of in vitro digestibility. Nutritional profiles also suggest that UALPCs, they have high amino acid content and low anti-nutrients. So some of the recommendations are that these LPCs have an appropriate yield, high protein content, desirable functional properties, high antioxidant activity with low anti-nutrients. They are certainly suitable to be used to supplement the diet and combat protein deficiencies. Therefore, we can say that UALPC has an edge over ALPCs as they are quite sustainable approach for valorization of agricultural byproducts to bioproducts in developing countries with promising health benefits. Thank you so much.